Hello everybody, here's Coronafighter campaign and we are here with you with another episode of webinar with expert with Dr. Mohtari Azad. She's a full professor of virology at School of Public Health in Tehran University of Medical Sciences, and she's also the director of the WHO National Influenza Center in Iran. Happy to have you here. Would you like to greet our audiences for a start, say hello something?
Thanks for your invitation. I hope that I provide some valuable information for your audience.
Would you like to elaborate on the COVID-19, its family virus, some specification, something that we can know about that?
COVID-19 is a new emerging disease that was recognized in late 2019. The virus which caused this disease belongs to coronavirus family that are enveloped RNA viruses with helical symmetry. This family has the largest genome in RNA viruses. Coronaviruses infected human and animals. The virus from, for example, birds or mammals cannot transmit directly from these animals to humans. Usually this family causing acute infection. Members of this family were isolated first in 1913 from chicken, but first human coronavirus was isolated in 1960 from a patient with upper respiratory symptoms. Until now, there are 7 human coronaviruses, which 4 of them cause mild respiratory diseases, but the other three ones like SARS, MERS, and this new virus that is named SARS coronavirus 2 are much more severe than and has more mortality rate. Our information about COVID-19 is developing day by day. The causative agent of this disease is genetically 80% similar to SARS coronavirus and 90% to bats. This virus infects both upper and lower respiratory and resulting from mild to severe lung disease, and may also infect other organs like kidney, heart, intestine. I might add that this disease is a serious disease for persons who are older than 65-year-old and has underlying disease like diabetes, hypertension, and coronary diseases.
How many times coronavirus has jumped the interspecies leap, and is this time any different or a new thing?
For the first time in 2003, we have a SARS coronavirus. After the emerging of SARS coronaviruses, that its origin was from bat, and after adaptation to palm civil, transferred to human. But fortunately, this virus has been eradicated, and no, we have not any case of SARS coronavirus in human. In 2012, we have another new emerging disease that called MERS or Middle East Respiratory Syndrome that transferred from camel to human. And now, in 2019, we have a new virus that causes COVID-19. So we have this phenomenon 3 times during the past two decades, and maybe we have in the future many viruses from this family.
Okay, are zoonotic viruses more potent pathogens generally, and if so, does time make them milder?
Most of the emerging viruses have origin from animals. Sometimes zoonotic viral diseases which are directly transmitted from animal to human could be very severe, but this is not a general rule. There is no evidence that by passage of time the virus became milder or its pathogenicity changed, but who knows, only times will tell us what will happen.
And I know that you have been in the charge of PCR tests here in Tehran University of Medical Sciences. How many PCR tests have you done so far, and to what extent can we trust these PCR tests?
Until now, it is about 15 days from the first cases of COVID virus in Iran. We have tested about 14,000 PCR tests in our lab, but the quality of result of the PCR test depends on the quality of the specimen and the time of sampling. We can rely on PCR tests about more than 70% because it depends the material that you use for your PCR detection. But I might add that until now we don't have any reliable serology test or antigen detection test for these diseases. So we should or we must rely on PCR tests.
Can we tell the time of first infection in a community based on the mutations and PCR tests you have done? Is there any data on what was the time that we had the first infection in our community, and what can PCR tests tell us about that?
After PCR, especially after conventional PCR, we can sequencing the genome of the virus and we can tell the time of the first infection in community. Actually, we need more equipped in our lab like next generation sequencing to sequence the whole genome of the virus to tell more information about viruses that are circulating in Iran.
Is there any possible target for medications against SARS-CoV-2?
I think I can tell that fortunately this virus has a specific enzyme like RNA-dependent RNA polymerase, helicase and proteases which are different from our enzyme. So there is a hope for a specific antiviral for this disease. So I hope that in very near future we will have a specific drug, antiviral drug for this disease.
And why do you think we haven't find any effective vaccine for COVID-19 so far? Is it something inside the virus that prevents us from making a vaccine to combat that?
Unfortunately, the coronavirus family don't induce a stable, prolonged, and productive immunity. And usually we see reinfection with this family.
Oh, yeah.
So it is very common we have reinfection from this family, just like rhinoviruses that causes common cold. Therefore, any vaccine cannot induce a long immunity more than wild type.
Interesting.
Therefore, we should emphasize more on developing antiviral medication. Of course, it is my personal opinion that antiviral may be more effective than vaccine.
Thank you, Dr. Mohtari, for your time and
Thanks
consideration.